And last video we have for chapter 17, or our inheritance chapter, before we move on to DNA, is genetics and society. So when we talk about genetics and society, we're talking about how things have changed over time. So one example of an organism that we have altered is corn. Corn is a big staple in our diet today, and it actually didn't exist on this planet originally. So it was actually selectively bred by crossing two different types of plants, tessonite and gamma grass. So they mated these two, or crossbred them, to create the corn that we do know today. Now, this did take thousands of years to get to where the corn is today. So it's hard to imagine that something like this created something like this. So selectively breeding does take a fairly long time. Canola is your next one. So this came from the rapeseed plant that was originally used as oil lubricant. The reason why they used it as oil lubricant was because it was considered, or it was, toxic for consumption. Now in Saskatoon, yay Saskatchewan, they bred a variety of these rapeseed plants that had very low levels of that toxic oil, so that they could make this oil more suitable for human consumption. So they selectively bred, or basically altered, the canola plant, or the rapeseed plant, so that we can actually use it for cooking. Animals we have altered, we've done this quite a bit. If you click the link, you'll see some very unique organisms, and they're quite funny, so go ahead and click that link. We have bred many animals to produce traits that we actually desire in them. So for example, cattle, we breed the cattle to produce more milk and have leaner meat. So we're going to mate those females that produce a lot of milk in hopes that their offspring will also produce a lot of milk. Chickens, so they want to mate the chickens that lay eggs with less cholesterol. How they figure that out, I don't know. And then just simply for pleasure, dogs. We breed dogs to a species exactly what we want. So Bichon Shih Tzu is my favorite dog. I have a Pomeranian Jack Russell Chihuahua cross. So we breed dogs to what we actually want in them. Now here's the meat of today's lesson, is pedigrees. So pedigrees are flow charts that are going to use symbols to show patterns and relationships, or the traits within the family over many generations. So this is like a family tree, more specific to a trait or a disease or a disorder. So what do the symbols mean? So you do have a data booklet. So your Science 30 data, Bio 30 data does have some of these symbols in it. So this is your Bio 30 data booklet. It's available online. It's also in Google Classroom under materials for quick access. So you can look through there if you don't have yours handy. So pedigree symbols. So it tells you a lot of the pedigree symbols in your data booklet. So males are represented by square boxes. Females are represented by circle. You never mix the two. Mating is represented by connecting a line between a male and a female symbol. Mating between close relatives is two lines. When you are representing the mating and production of offspring, you put the offspring in the center of the mating line, and then you create your offspring. So you'll notice here that generation 1 and generation 2 are nice, neat lines. That's also how you create it. So you're not going to have one generation person here, and then one up here, and then a line going from here to way up here. Okay. Identical twins. Okay. Non-identical twins. Affected individuals are completely shaded. Some diagrams will show them known as heterozygous, so they're half shaded. Or they can be carriers, for example. No carriers of an X-linked recessive trait. You have a dot in the middle, deceased individuals, and then sex unknown. For the pedigrees that you guys are going to be working with, you're going to be working with a lot of male-female mating, generations, and then affected individuals. I haven't really seen a lot of the heterozygotes like this, but you could potentially. So, autosomal inheritance. Autosomal inheritance refers to traits that are coded by genes on the autosome. So these are the non-sex chromosomes. So we have 23 chromosomes, 46 total. Okay, we have 23 pairs. Okay, the 23rd pair is considered your sex chromosomes, your X and Y. So everything else, 1 to 22, is autosomal. Okay, so any gene located on a chromosome 1 to 22 is known as an autosomal trait. So, examples. So I'm not going to go through all of these in detail, and I'm going to give you a practice sheet that I want you to practice. I'll post the answer key so that you can make sure you're doing it right. So the following throws a 3-generation pedigree for the autosomal dominant trait. So right away it says autosomal dominant for polydactyly, right in the genotypes and phenotypes for every individual. Okay, so I know it's 3 generations because I have 1, 2, 3. So we have grandparents, parents, these were married in, and then we have grandkids. Okay, this is autosomal dominant, which means that it's going to be, anytime you have a big letter, it's representing the trait of polydactyly. Okay, looking at parent 1 and parent 2. They both have polydactyly, which is telling me they could be big B, or big B, little b. We won't know until we look at their offspring. Okay, so looking at their offspring, we have 2 offspring that had polydactyly and then 1 that didn't. So I'm going to do this. Polydactyly, little b, normal. Okay, so I know that this person right here, number 4, has to be little b, because they're not shaded in. They have normal fingers and toes. So 10 fingers, 10 toes. Okay, so in order for this offspring that came from 1 and 2 to get a little b had to come from mom. Sorry, mom's over here. And a little b had to come from dad. Okay, so we know that they have to have a little b. They can't have 2, because if they had 2, then they wouldn't be shaded in, which means that the parents have to be this. Okay. Now, if I cross these 2 individuals, okay, and you should be able to, with the Punnett squares that you've done before, know what your ratio is. You should know that you get 1 big B, 2 big B, little b's, and then 1 little b. Okay, now again, this is probability. It's not saying you get 1 of these, 2 of these kids, and 1 of these kids, because we don't know how many kids they're going to have. So if I look, these 2, when I cross them, you can see that definitely for number 4, you do get the possibility of being normal. 75% chance you are not normal and you do get polydactyly. No, I'm not saying it's not normal, but it's different, right? So I have to figure out what 2 and 3 are. Okay, let's start with 3. 3, could they be this person, big B? Yes, because they're infected. Could they be big B, little b? Yes, because they're infected, or they have polydactyly. Do I actually know? No, I can't tell because I can't see what their kids are. So I have no proof of what they are. So that means they could be big B, or big B, little b. So you put big B with a line saying that it could be big or little. Now looking at person 2, okay, in generation 2, okay, I know that the male that she mated with is little b, little b. Person 1 down in generation 3 is little b, and person 3 is down here. Okay, anybody that's not shaded is little b. So I know all of these guys are little b as well. Okay, so I have to figure out what number 2 is. Okay, so I know Dad is going to only ever give little b, or a little b. Okay, I do have children that do not have the trait. Dad can only pass on one of these genes, which means in order to get 2, Mom had to have a little. Okay, since she has the trait, she still has to have a big. So big, little, 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 big, little, little, little. So Mom had to be big, little. There was no choice in the matter. Now, number 2 and 4. Do I know what they are? Well, they don't have offspring, but if I look at parents, can you ever get a big, big? No, because Dad is little. So, out of this combination, these guys have to be big, little. So, big, little. Little, 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 little. I feel like I'm making 6s. So this is representing my pedigree with Punnett squares. So now we're combining everything. I want you to try number 2 on your own, okay, and then check with the answers to see that you've done it right or pretty darn close. The other type of inheritance is sex-linked inheritance. So this is referring to any genes that are located on the sex chromosomes, so the X or the Y. Now again, most traits are found on the X chromosome. Okay, so the following pedigree, sorry, I can't talk, shows the inheritance of hemophilia. An X-linked recessive, Control Z, get rid of that. I want my highlighter back. An X-linked recessive trait. So it's found on the X chromosome. That is what it's telling you. Note, only the offspring that are either affected or are carriers are shown in the pedigree. So that's telling you that this pedigree is not a full pedigree of the family. That's what it's saying. Write the genotypes and phenotypes of individuals. Okay, so remember back to the beginning, the dot represented a carrier of an X-linked recessive trait. We do have an X-linked recessive trait of hemophilia. So this means when I have sex-linked, I have to have XXXY included. Okay, so this is a female, so I know that this is going to be XX. Now she is a carrier, which means she has the trait, but she's not expressing it, which means she doesn't have hemophilia. Since it's recessive, she needs to have a big B as well to show that she doesn't have the trait. Prince Albert is not affected, so affected will be completely shaded in. So he is XY. Nothing is found on Y because it's an X-linked recessive. So that means on there, his X chromosome, he has to have a capital B. Okay, moving down. So Victoria is a carrier, so exactly like her mom. X big B, X little b. Frederick is not affected, so he's like Albert. So we have X big B, Y. We're going to look at their offspring. Waldemir died at age 11. It's hard to show that that's crossed off because it's shaded in. So X little b, because he's affected, and we have Y. So this one's pretty self-explanatory, so I'm not going to finish this off, and I don't expect you to either, because this one is fairly easy because it shows you the carriers and then the non-carriers. So these are a bunch of genetic disorders. All of the ones that have the asterisks beside are ones that you actually had to memorize for tests. But since you can go back to your notes package, you can look at it. But if you were writing your diploma, you would have to memorize these ones, including hemophilia up there as a sex-linked X recessive. So all of these you can read through. I'm not going to read them for you. You can read through them just as well as I can. Okay? So why, oh, why do we want to know this? So if you had a genetic disorder, would you want to know? Would you want to know if your unborn child is going to develop your genetic disorder? So this was more of a discussion kind of question that we would discuss as a class. Unfortunately, we are where we are in the world today. There is no right or wrong answer to this. If you were to know that if you had a genetic disorder, would you have kids? It is totally your choice. Okay, there's nothing saying yes, you have to, or no, you don't have to. Okay, it's totally up to you. So, moving on to genetic screening. So, genetic screening allows geneticists, so scientists that study the human genome, so the karyotype, can determine who is at risk for developing certain genetic conditions or passing these genetic conditions onto their children. So genetic screening can be done by using chronic villi sampling, so the sampling of the villi that during pregnancy, or can be done by doing an amniocentesis. So both of those were discussed in the reproduction unit. Sorry, forgot it for a second. So they can use those. You can do a blood test to extract your genetic information from that, but generally they want to do these to determine if your child, your unborn child, is going to carry a genetic disorder. Genetic counselors can estimate the risk of inheriting a particular genetic condition and can inform you of those symptoms or treatments for certain diseases. Okay, I am going to also post the pedigree analysis problems. So I kind of put hints on the front page to how to tell which type of pedigree it is, because you on a test may be given a pedigree and you have to determine, is it X-linked, is it X-linked recessive, is it autosomal dominant? So you can use this as a guide. Okay, and then there's practice questions on the back. I highly recommend that you try these and then check the answer key to make sure you're doing it right. Good luck.